everybody. In this last week of the course on supramolecular chemistry, we are going to look at some important applications. And to start with, we are going to explore the applications of the photophysics associated with supramolecular systems. So, in the previous lectures, we have looked at the different photochemical procedures where the excitation is brought about by light and then organic transformations were achieved. Now, we are going to look at photoexcitation processes which lead to different photophysical processes which bring about changes in the luminescent profiles of the samples. And we can have both radiative process as well as non-radiative processes. So, when you have photoexcitation, we have looked at this part a little bit initially. So, in photoexcitation, we can have different processes. We can have photophysical processes, we can have photochemical processes. This photochemical processes can lead to chemical reactions in the excited state. For example, secondary charge separation, isomerization reaction and dissociation. These kind of photochemical reactions can take place in the excited state where a charge can be transferred from the donor molecule to the acceptor molecule, as has been observed also in the process of photosynthesis. We can have trans-cis isomerization in the presence of light, and we can also have dissociative processes such that the weak bonds in the molecule can break. In the photophysical properties, we have looked at the concept of fluorescence and phosphorescence which are luminescent procedures, which are luminescent processes. But we also can have these are the radiative processes, and we also have the non-radiative processes, which are the electron transfer and the energy transfer processes as well. Okay. So, what is important is that we can have either the fluorescence and phosphorescence processes. These are of importance in sensing and signaling, whereas the chemical reactions of the excited state, the photochemical processes, are important in the fields of application to molecular switches and photocatalysis. Molecular switches and photocatalysis. So, we can also now look at how this process of energy transfer is important, because we are also going to look at the applications of these photophysical properties in molecular wires. And before we go into the applications of molecular wires, we will see how the process of energy transfer in a non-radiative process, or we can call it the non-radiative process, which is the non-radiative transfer of excitation energy before the sample can undergo luminescence by removing the excess energy in the form of photons of reduced frequency. So we have this energy transfer process, which is actually non-radiative process, which involves transfer of the excitation energy by non-radiative means. So this energy transfer mechanism operates via Forster type, which is the electrostatic interactions between the donor and the acceptor. These are the electrostatic interactions between the donor and the acceptor. And the number 2 is the Dexter type, which is the exchange energy of the electrons. So we can look at these processes more carefully now. So say we have got the excited state of the donor, this is the HOMO and this is the LUMO. So we have now promoted the electron and it now interacts with the acceptor, which is in the singlet state, and there is now a singlet to singlet Forster energy transfer. So this will now de-excite and this will now undergo photoexcitation. So this comes back to the ground state and then this goes into the excited state. So you have this kind of a Forster energy transfer from the donor to the acceptor. So we can say a D star plus A going to D plus A star, where the spin multiplicity is the same for the donor and the acceptor. We can have the process via the, so this is the Forster energy transfer. We can also have the Dexter energy transfer, where we have the photo excitation, that is 1 D star, plus now we have both these electrons 1 A, and now there is an exchange. So this electron goes here and this electron goes here. So there is a electron exchange, and this exchange process gives rise, this is star, to the ground state of the doublet, plus, so we have now in this case we will have to put this slight correction here. It is has to be of the opposite spin, so this will go here. So this will be now so here we have got, so it is this one which goes, and therefore we have got this one and this one. So this is now the 1A star. So this is the singlet to singlet Dexter energy transfer. And we can also have another case where we have the triplet excited donor, that is these are of the same spin orientation, that is a triplet state, and this interacts with now again the acceptor. So this goes here, this is exchange, there is an electron exchange now happening of the, this is the triplet to triplet Dexter energy transfer. So this will lead to the formation of the ground state of the donor and the triplet state for the acceptor, which is in the excited state. Okay, so this is the accepted, this is the donor triplet, this is the acceptor triplet. So this is a triplet to triplet Dexter energy transfer which can operate. So in all these processes there is a transfer of energy without the release of photon. In the Coulombic it is purely electrostatic, and the extent of energy transfer depends upon the separation between the donor and the acceptor. and it actually occurs at very short distance separations, the efficiency of the process, and overall these processes lead to quenching of the luminescent processes. For example, it leads to luminescence, leads to quenching of fluorescence or phosphorescence in the materials of interest. So, we can actually look at some examples to understand these photoexcitation processes more closely. So, for example, we can look at electron transfer processes, and we can also look at energy transfer processes, in covalent and non-covalent systems. So, for example, we can take this example. Where we have a zinc porphyrin chromophore. So, we have a zinc porphyrin chromophore, so this is the zinc 2 plus, and here it is connected to the 5-membered ring, and here I am again drawing so we have this one, this one, this one, and this one. This is iron chloro bond. Okay. So this is iron in the 3 oxidation state, and we can now compare it with, so this is a covalent backbone, this is connected by a covalent backbone, the 2 chromophores, this is a zinc porphyrin and this is the iron 3 chromophore, and we can compare the same system now I am not drawing it again. We can see this particular arrangement where it is connected via a non-covalent backbone. So, so they are non-covalent interactions, purely hydrogen bonded, and these are modular components. These are modular components. The zinc 1 as well as the iron 3, and on photoexcitation, now when there is H nu and there is photoexcitation, there is electron transfer to the iron 3 center, which leads to the formation of the iron 2 center. And so this electron transfer processes which happen here on photoexcitation is responsible for creating the charge separation between the donor and the acceptor. So now there is electron transfer from the donor to the acceptor, and this electron transfer takes place either via the covalent backbone or it takes place via the non-covalent backbone. And this electron transfer processes are quite efficient in the sense that the rate of photoelectron transfer, if I were to consider this A and this system as B, it is 4.3 into 10 to the power 9 for B, and it is 8.1 into 10 to the power 9 for A. So you can see that even the supramolecular motif is as efficient as the covalent backbone in transferring the electron from the donor to the acceptor, and therefore creating the necessary charge transfer, which we refer to as a secondary charge transfer. And these electron transfer processes are important in different applications. For example, electron transfer processes are important in photosynthesis and also in the design of diode materials. So, so we see that this electron transfer process takes place in a very efficient manner. Similarly, we can also take an example where we can look at a system where we can look at the energy transfer process. Where we consider okay, and here if when we interact with bi-napthyl, this is in anthracinyl moiety, then in the presence of an acid and in the presence of a base. So here what do we happen? This gets protonated, and then there are hydrogen bonds. It forms with the ether, and now you have got this ether moiety, and you have got the bi-napthyl here, and you have got the anthracinyl here. So when you do the photoexcitation here, H nu, this is followed by energy transfer. This is the non-radiative energy transfer from the bi-napthyl unit to the anthracinyl unit, and this leads to luminescence of the anthracinyl moiety, where it quenches the fluorescence of the bi-napthyl moiety. So in the absence of this complexation or supramolecular hydrogen bonding association, what will happen is that the bi-napthyl will take place, but that is quenched because of this complexation, and the energy transfer process now happens from bi-napthyl to anthracinyl, which now undergoes releases for light of photon of energy h nu prime, and that is responsible for the luminescence of anthracinyl solution. And it is very sensitive process, because if we now replace the methyl by phenyl, then no complexation takes place because of the steric mismatch. So with this background, we can now go and look at the application of this in molecular wires. We can take up an example in molecular wire, and so generally the concept is that you have got a donor and you have got an acceptor, and there must be a pathway for electron transfer or conduction. This process must take place, and this process has to happen in a rapid way, because electronic flow is the opposite of electronic flow is current flow. And to start with, Jean-Marie Leigh's group have addressed this problem by the preparation of a large number of carotenoid complexes, which he calls as carovialogens. So these carovialogens have been made. Let us look at one particular example of this carovialogen. We have the protonated, the methylated pyridine. So, and the thickness is comparable to the length of the wire. So this particular carovialogen has been formed. It contains alternating alpha single and double bonds, and therefore this has been observed to function as a molecular wire. And interestingly, you can see that these are the hydrophilic groups, and this is the hydrophobic backbone. The hydrophilic groups and the hydrophobic backbone. And because this has got both the polar and non-polar parts, it is something which is of relevance in transfer of signals in cells also across the bilayers. So if there has to be a signaling mechanism between the intracellular and the outer and the extracellular region via these bilayers, then the bilayers have got both the hydrophobic part as well as the hydrophilic part, and this particular example serves to illustrate that electron transfer processes or current flow is very important in the biological path, in understanding different biological pathways. So in the next class we will now look at little bit more detail into the functional aspects of the system as molecular wise. Thank you.